Today we will understand Module 3 of Chapter 16, Heredity and Variation. In the previous module, we have studied about deoxyribonucleic acid, that is DNA, gene, and ribonucleic acid, that is RNA. In this module, we will study about Mendel's principles of heredity, Mendel's experiment of the monohybrid cross, and Mendel's experiment of the dihybrid cross. Students, Gregor John Mendel was an Austrian scientist. He was born on 20th July 1822 and died on 6th Jan 1884. He studied the inheritance of some characteristics of the pea plant. He showed that inheritance of these characteristics follows certain principles. Later, these principles became popular by his name. Mendel's work was recognized only in the 20th century. Now let us see the Mendel's principles of heredity. Students, genetic material is transferred in equal quantity from parents to progeny. Principles of heredity are based upon this fact. If both the parents make equal contribution to inheritance of characteristics, which characteristics will appear in the progeny? Mendel carried out research in this direction and put forth the principles of heredity responsible for such inheritance. Students, the experiments performed by Mendel almost a century ago are quite astonishing. All of Mendel's experiments were based upon visible characteristics of pea plant, that is Pisum sativum. These characteristics are shown here. Shape of the seed, that is round or wrinkled, color of the seed, yellow or green, then color of flower, purple or white, shape of pod, inflated or constricted, color of pod, green or yellow, position of flower, axillary or terminal, and height of plant, that is tall or dwarf. Now, let us see Mendel's monohybrid cross. Students, in this experiment, Mendel brought about the cross between two pea plants with only one pair of contrasting characters. This type of cross is called a monohybrid cross. I repeat, in this experiment, Mendel brought about the cross between two pea plants with only one pair of contrasting characters. This type of cross is called a monohybrid cross. So, as to study the monohybrid ratio, let us consider the characteristic plant height with a pair of contrasting characteristic tall plant and dwarf plant. So students, Mendel referred to the tall and dwarf plants as dominant and recessive. I repeat, tall pea plants and dwarf pea plants were used in this cross. Hence, this is a parental or parental generation, that is P1. And in this, Mendel referred to the tall and dwarf plants as dominant and recessive respectively. The tall plant was referred to as dominant because all the plants in the next generation were tall. I repeat, the tall plant was referred to as dominant because all the plants in the next generation were tall. The dwarf plant was referred to as recessive because this characteristic did not appear in the next generation at all. The experiment has been presented by Punnett square method as shown. Students, Punnett square is a diagram that can be used to predict the genotype and phenotype resulting from genetic cross. Students, according to the Mendel's law of segregation, only one of the two gametes, as you can see here children, the parental generation, that is TT, capital TT and small tt, represented for tall and dwarf plant. So, according to the Mendel's law of segregation, only one of the two gene copies present in an organism is distributed to each gamete. Only one. Okay. That is egg or sperm cell that it makes, and the allocation of gene copies is random. When an egg and a sperm join in fertilization, they form a new organism whose genotype consists of the alleles contained in the gametes. So this is how the gamete formation takes place. I repeat, according to the Mendel's law of segregation, only one of the two gene copies present in an organism is distributed to each gametes, egg or sperm cell that it makes, and the allocation of the gene copies is random. When an egg and a sperm join in fertilization, they form a new organism whose genotype consists of the alleles contained in the gamete. So this is how the gamete formation take place. So we get a gamete in the parental generation from TT, capital TT and small tt. We get one of the gamete, that is T and T, and we by the cross of it we get capital T and small t, and in the F1 generation, when the mating of both is been done, we get a ratio of 1:2:1 of tall and dwarf as 3:1. Students, depending upon the observations, Mendel proposed that the factors responsible for inheritance of characteristics are present in pairs. I repeat, depending upon the observations, Mendel proposed that the factors responsible for inheritance of characteristics are present in pairs. Let us say the first filial generation, that is F1. In this experiment, Mendel observed that all plants of first filial generation were tall. They are represented as capital T, small t, capital T and small t. But I realized that tall plants of F1 generation were different from the tall plants of P1 generation, because parents of F1 tall plants are tall as well as dwarf plants. Depending upon observations of F1 generation, Mendel concluded that factors of tall plants are dominant over the factors of dwarf plant. Though all the plants in F1 generation were tall, they contained the factors responsible for dwarfness. That is, though the phenotype of F1 plants is tall, their genotype is mixed. I repeat, though all the plants in F1 generation were tall, they contain the factors responsible for dwarfness. That is, though the phenotype of F1 plants is tall, their genotype is mixed. Students, phenotype means external appearance or visible characteristics of organisms. For example, tallness or dwarfness of plants. Whereas genotype means the pair of genes, that is factors responsible for visible characteristics. I repeat, phenotype means external appearance or visible characteristics of organisms. For example, tallness or dwarfness of plants, whereas genotype means the pair of genes, that is factors responsible for visible characteristics. Students, genotype of P1 plants, P1 tall plants is capital TT, produces only one type of gamete, that is T. Genotype of F1 tall plants is capital T small t and it produces two types of gametes, capital T and small t. We can see that in case of tall plants of F1 and P1 generation, though they show similar phenotype, their genotype are different. Mendel further continued this experiment and brought about self-fertilization in F1 plants, from which a second filial F2 generation was produced. So second filial generation, that is F2, in the second filial generation F2, both tall and dwarf types of plants appeared. According to the data collected by Mendel, out of 929p plants, 705 were tall and 224 were short. The phenotype ratio of this is 3:2, that is 3 tall and 2 dwarf, and genotype ratio is 1:2:1. It can be inferred that in the F2 generation, phenotypically there are 2 types of plants, whereas genotypically there are 3 types. These types are shown in the table. F2 pure dominant, that is capital TT tall plants, homozygous. Students, homozygous means having 2 identical alleles for a particular gene. See here, having two identical alleles for a particular gene. It is called as homozygous. F2 pure recessive small tt dwarf plants, again homozygous. Then F2 hybrid plants, capital T small t tall plants, that is heterozygous. Heterozygous having two different alleles for a particular gene. Now let us move to the Mendel's experiment on dihybrid cross. In the dihybrid cross, two pairs of contrasting characteristics are under consideration. Mendel performed more experiments on hybridization in which he considered more than one pair of contrasting characteristics. Mendel brought about a cross between a pea plant producing rounded yellow colored seeds and a pea plant with wrinkled and green colored seeds. In this cross, two pairs of contrasting characteristics were considered: color of seeds and shape of seeds. Hence, it is called a dihybrid cross. I repeat, in this cross, two pairs of contrasting characteristics were considered: color of seeds and shape of seeds. Hence, it is called a dihybrid cross. Let us see the P1 generation, students. That is parental generation. Mendel selected the pea plants producing rounded yellow seeds and wrinkled green seeds as parent plants as shown. Students, so phenotype, that is rounded yellow seeds and wrinkled green seeds. Here is a cross between two pea plants, one with yellow round seeds, that is RRYY, and one with green wrinkled seeds, that is small RRYY. Because each parent is homozygous, the law of segregation tells us that the gametes made by the wrinkled green plants all are small ry. The gametes made by the round yellow seeds or plant are all capital RY. Thus gives us F1 offspring that are all rounded yellow seeds, that is capital R small r, capital Y small y. Students, the allele specifying yellow seed color is dominant to the allele specifying green seed color, and the allele specifying round shape is dominant over to the allele specifying wrinkled shape as shown by capital and small letter. So this is how the gametes are formed. This means that the F1 plants are all yellow and round because they are heterozygous for two genes. That is, the F1 plants are called dihybrid. When Mendel did this cross and looked at the offspring, he found that there were four different categories. As you can see here, four different categories of pea plants: yellow and round, yellow and wrinkled, green and round, and green and wrinkled. So these are the different gametes from as shown in the previous diagram. By selfing in the F1 generation, we get four different gametes: RY, capital R, small y, RY, and RY. Students, during gamete formation in P1 generation, the pair of gametes separate independently. That is, in RRYY plants, only RY type gametes are formed and not RRYY. Similarly, in small RRYY plants, only small RY gametes are formed. Thus, we can say that each pair of genes is represented in the gamete by only one gene from that pair. Based on the conclusion from the monohybrid cross, Mendel expected that in the F2 generation of dihybrid cross, plants would produce rounded yellow seeds. Though the genotype of these plants was capital R, small r, capital Y, small y, their phenotype was like the parents producing rounded yellow, because yellow color is dominant over green and round shape is dominant over wrinkled. Due to the combination of two different characteristics in the F1 generation of dihybrid cross, these plants are called dihybrid plants. Plants of the F1 generation of dihybrid cross produce four types of gametes, as I told earlier also: RY, RY, small ry, small r, capital Y. Of these gametes, RY and RY are similar to those of the P1 generation. As you can see here, this RY and small ry, capital RY, small ry are similar to the P1 generation. So this was the Punnett square diagram. Arrange, gametes are arranged, that is the male gametes and female gametes. And after crossing, we get about 16 gametes here, 16 seeds. So the ratio is here, round or yellow. If you see here, 1, 2, 3, 4, 5, 6, 7, 8, 9. After crossing RY and RY, in this way we get 9 seeds. Round or green, 1, 2, 3. Wrinkled or yellow, again 1, 2, 3. Wrinkled or green, that is 1. So phenotype ratio is 9:3:3:1. Let us understand points to remember. Genetic material is transferred in equal quantity from parents to progeny. In the Mendel's monohybrid cross, Mendel observed that all plants of first filial generation were tall. Plants of the F1 generation of dihybrid cross produce four types of gametes: RY, RY, RY and small ry. There is a short homework for you. Explain Mendel's monohybrid progeny with the help of any one cross. Explain Mendel's dihybrid ratio with the help of any one cross. Distinguish between monohybrid cross and dihybrid cross. Thank you.